The overall goal of this experiment is to create an organotypic model that mimics the peritoneal microenvironment to better our understanding of ovarian cancer biology.
This method can help answer key questions in the ovarian cancer field, such as how do interactions between cancer cells and the microenvironment contribute to disease pathogenesis, and how can potential inhibitors of these interactions be tested. The main advantage of this technique is that actual primary human cells that form the mesothelium-lined surfaces of the peritoneal cavity are used in the ovarian cancer cell co-culture.
Though this method can provide insight into ovarian cancer, it can also be applied to the study of other cancers that disseminate throughout the peritoneal cavity, such as gastric, pancreatic, and colon cancers.
Upon acquisition of a surgical human omentum specimen, immediately immerse the sample in room temperature PBS. Within 2 hours of the immersion, transfer the omentum to a 50 milliliter conical tube containing 20 milliliters of fresh PBS. Transfer the remaining PBS wash containing the primary human mesothelial cells, or HPMC, and red blood cells into a new 50 milliliter conical tube and spin down the cells. Then pour the tube contents into a 10 centimeter sterile culture dish and use 2 scalpels to mince the tissue into 5 cubic millimeter pieces. To isolate the primary human mesothelial cells, or HPMC, transfer the omentum pieces into a 50 milliliter conical tube and wait 1 minute for the solid pieces to float to the top. The HPMC will settle at the bottom.